Okay, so we have first to go to the question. I think that the sample size is getting small and small. When you see 30%, 30%, 30%, it's because there's two people in the room who vote for that. Okay, but nevertheless, the questions are very interesting. To select a particular DCB in your clinical practice, you mainly consider the available scientific evidence. 100%, one vote. Your personal experience, 2 vote, 100%. The price, 4 vote, 100%. Others. So that's the final word. The available scientific evidence and personal experience, 8 people. Other, 9 people. Be careful. 1 time. 2 time. Adjudicate. 9 people. Okay? So the answer is the available scientific evidence. I think that's what we all do. Of course, the order is the quality of the marketing. How nice is the lady or the man which is coming to sell the product. That plays a role, but otherwise we look at the scientific evidence.
Yeah, and maybe also the trackability and things like that may count in some cases. So it's not only the scientific evidence, but it's most important.
Sure. Small vessel, diffuse, you have to get there. Of course, if somebody start to do proximal LED and mainstem, it might be somewhat less relevant. Should we go to the next question? Because I'm sure that Juan Granada is waiting. So the second question is regarding commercially available Limus DCB. Are my first choice. I never use Limus. Are my first choice. I never. How does it work? There is question and answer coming. Okay, there is a 6 vote. Paclitaxel DCB should be preferred. 6 case, 7 case. I never use Limus DCB. That's 14%. That must be 1 person. And yeah, I will not calculate the power of that study. Okay, but basically many people believe that paclitaxel should be preferred. I think that so far the strong data on Limus still have to come. I mean, everybody is waiting for of course, things like Transform 2 and the solution study, and that would be good. Okay, if there is no interaction from the panel on the first and second question.
But this is the new one, no? This is a new one.
That's another one, but about the first 2, I mean, anything you would like to say?
Maybe. Not the first. Maybe it depends on the availability on the cath lab also, the one that I choose, the sizes, or even if we are in a clinical trial, because we used to do a lot of, so it could be related also with the second question about what I prefer. I think that I feel much more confident with paclitaxel because I have used it much more, but even Limus we have to see.
So certainly, yeah.
It was surprising that 1 person voted Limus is my first choice. I would like to know what's the reason to choose Limus as first option.
There is a privacy law in Europe. So, but the person who is courageous, no? Yeah. Okay. Okay, let's go on then. What percentage of your PCR will be using DCB from no on? 9, so it's 4 versus 5. Yeah. I think that the country who use a lot of DCB is Japan and China. I think the numbers that I've seen is 20% for the whole country. You have the paradox that in China, but also in Japan, the drug-coating balloon cost more money than the drug-eluting stent. Price has been imposed in India and has also been imposed in China. There was even worse. There was a tender in China. So some of the stents were put on the side, and the government went to the first generation of the stent, not the second generation. So it was really, but it seems that now it's going to be corrected.
So may I ask your opinion, what is the reason for high use of DCB in Asia? Is it because they have smaller coronaries and or a lot of diabetes? What do you think?
Right. I mean, there is a lot of diabetes. There is also for the Far East that you don't want to alter your body. You don't want to place a piece of metal. So that's a very strong argument. They were already buying, I would say, the biodegradable scaffold because it was not a permanent foreign body, but this is the same for the drug-coating balloon. That was probably the last question. Yeah, correct. And let's check if Juan Granada is there. Yes, at least his slide. We see your face on the side. It's perfect. How are you?
Doing great,
Good.
great, Patrick, and I'm enjoying a lot the conversation. Actually, very interesting discussion and very interesting questions as well.
Yeah, I must say that the whole day was made of paradox and pro and contra, and I think it's, I don't know any technique in interventional cardiology where it doesn't take 10 years to have some idea how to do it. You know, we did that for atherectomy, rotablator, drug-eluting balloon. Don't forget that was before the stent. It always costs 10 years. 10 years how to do it. Juan Granada, we are listening to you. It will be, I'm sure, very expecting with lesson learned, remaining challenge and future perspective. Go ahead.
Patrick, and thank you again for the introduction. Fernando, thank you for the kind invitation, and I really hope I can really make it in person to the next version of the meeting. You know, I was listening carefully about the previous discussion and the questions that you asked, and you know, I really think that this talk is going to at least provide some perspective, you know, in terms of the lessons that we learned, the remaining challenges, and the most important thing is why is that important for the future technologies that are coming into the market, hopefully very soon. My disclosure, I don't have any personal conflicts. All are institutional conflicts with the companies we work with. So I have to say that this is a field that has not really been easy to be part of. And I have to tell you that the people that continue to be involved, I have to just congratulate, because I feel like this is a group of renegades, you know, people that continue to believe in the concept of leaving nothing behind, of doing a smart angioplasty, and actually getting better outcomes for the patients. But I pulled this slide, I went back to my slides, and I found the slide in 2019, you know, in a talk that I gave at TCT, when we actually were even actually debating about the name, and debating actually even about the possibility that this technology had a chance to work. I mean, you see the bottom part of it says the type of technology is a huge leap of faith. This is actually something that I heard from some, you know, people involved at that time in drug-eluting stents. And I have to tell you that actually took a lot to be able to get where we are right now. The most convincing data was generated at the very beginning in the peripheral vascular territory, primarily driven by the fact that multiple concepts were developed, you know, fueled by a lot of money that actually went into innovation, and multiple concepts and devices were developed. And at least one thing that is very important to say is that early on, the biological effectiveness and safety profile of paclitaxel coated balloons, you know, was firmly established. Small randomized studies, but the consistency of the data essentially gave a lot of comfort, you know, to the public and to the investigators, that this technology actually had a role in interventional cardiovascular medicine. Then we moved into a phase in which everybody started to ask, okay, so if this is going to work, I mean, a single delivery of a drug, how does this work? And I think preclinical data translational research help us to answer a lot of questions. Important actually lessons that actually we learn and are very important for the new generation drug-coated balloons is the fact that actually we established the importance of using a carrier in balloon-based drug delivery. This is particularly important for sirolimus compounds, more than actually paclitaxel compounds. We were able to establish that the coating essentially fragments and adheres to the vessel surface, and these actually biological depots are responsible for the long-term release. We actually were able to establish that the chemistry of the particle that essentially is attached to the vessel wall is very important and determines the pharmacokinetics of the balloons. Highly insoluble particles produce, a very long-term release. Highly soluble particles that we call amorphous coatings produce very fast release and actually shorter retention rates. We also were able to establish the importance of dosing. Assuming similar paclitaxel coating characteristics, actually dosing does matter. And you see in experimental studies, we were able to show that if you actually go from 3 micrograms to 1 microgram using the same coating, you reduce efficacy by approximately 50% in terms of neointimal proliferation. So the concept of dosing has to do not only about the concentration, but actually also has to do with the chemistry of the particles that are adhered to the vessel wall as well. We were able to actually convince a lot of people that it was a biological mechanism that was plausible and was responsible for the good clinical results that paclitaxel-coated balloons were having. But then we enter into a panic phase, where we started essentially describing the mechanism of action, the fragmentation of the coating, and the fact that it produces particles as part of the mechanism of delivery. We were able to establish the different percentages of particle actually dislodgement, and that this was actually part of the mechanism of action of these balloons. And one of the biggest questions that linger around for several years is what was the biological effect of long-term paclitaxel tissue residency in wound healing, or actually distally in the myocardium after you dilate, these balloons. We essentially did a lot of studies, Renew did a lot of studies, and many investigators did a lot of studies in which we demonstrated that in fact, actually paclitaxel is found in the tissue distally where the balloon was inflated. This is a very elegant study that we did inducing actually ulcers, distally in healthy animals and balloon, the proximal vascular segments. And we were able to demonstrate that actually the paclitaxel levels were so small and so scattered that did not really affect, wound healing in these healthy models. Again, these are healthy models with no vascular compromise, but it was a very nice way to show and demonstrate the safety of the studies. Renew did a lot of studies in the myocardium as well, demonstrated that the amount of particles were small, covered actually less than 0.1% of the myocardium, and really did not affect actually the tissue in general. Then in 2018, a nuclear bomb was essentially dropped into the field with the meta-analysis presented by Cazzano. I'm not going to go into the data or the reasons, but the reality is that actually took us a long time, at least a year, to get the FDA aligned to the facts and actually the data. And right now, I think the field was able to recover after multiple meta-analyses, including a complete follow-up of patients, essentially showing that these apparent differences in all-cause mortality were not significant between paclitaxel balloons and controls. So then, during that phase, we went into an aggressive phase of drug-coated balloon innovation. This was an opportunity. We had first-generation coatings that actually worked, but had some safety concerns. And as happens in our fields, always, this creates significant opportunities for innovation and development of new technologies. And the field actually moved from highly crystalline coatings that essentially were very effective, but had a high amount of variability and inconsistent tissue levels, to more actually organized coatings, using different strategies, decreasing the solubility of the particles, controlling the crystallinity, or actually encapsulating paclitaxel, all aiming to decrease the perceived potential toxicity of paclitaxel in the vessel wall. And this is when the sirolimus story essentially started to come in. And this is actually in a way biased by our history in drug-eluting stents. I am not going to debate that from the safety and efficacy profile, sirolimus has by far a much better biological profile than paclitaxel. But I feel that actually for drug-coated balloons, the field forgot one important thing, and is the pharmacokinetics of the drug. Paclitaxel has a fast tissue absorption and very long tissue retention. Sirolimus has a slow tissue absorption and very short tissue retention. So for balloons, what is actually good for stents is not really good for balloons, and the other way around. Paclitaxel for this particular purpose is actually much better suited for balloon delivery than sirolimus. That's the reason why we started to talk about the design characteristics that were needed for a sirolimus balloon to actually work, which are different than paclitaxel-coated balloons. Drug transfer is inefficient from balloons, so a carrier or a delivery method was actually required. Tissue absorption is slow following the acute drug transfer, and degradation occurs, so drug protection is actually really needed in order for sirolimus to work, and the half-life is actually relatively short, and the biological effect depends on maintaining therapeutic levels, so sustained and controlled drug delivery is actually favored. A very different type of technology compared to paclitaxel-coated balloons. That's the reason why all balloon technologies using sirolimus right now have some level of encapsulation to be able to protect the drug and being able to maintain the tissue levels over time. But again, the pharmacological profile is very different compared to paclitaxel products. So this is actually my take on sirolimus, and I would actually say it's a word of caution without actually having clinical randomized data right now, is that sirolimus-coated balloons were developed under the assumption that paclitaxel was both more toxic and less potent compared to limus drugs. And I really think this is actually true in the world of stent-based drug delivery, especially in the presence of a polymer, and I think we're all convinced of that, and the data is very clear, with the stents. But the potential toxicity of paclitaxel is greatly minimized when delivered in the absence of a polymer, in this case, balloon coating. The long-term anti-restenotic effect is determined by the sustained presence of anti-proliferative drugs. In this regard, paclitaxel is better suited to achieve this biological effect when delivered via balloon coatings. I'm not really saying that sirolimus is not going to work. What I'm saying is that if you ask me, technologically speaking, if I had to start a company in drug-coated balloons, I would actually say paclitaxel is a much better drug for this particular application. Now it comes down to technology adoption and competitive landscape, because we're introducing these technologies in a field that is crowded, that is changing, that the indications are actually evolving, and where all this information really is going to fit in the future. And this is a question that actually I get a lot from many people, especially startups. How can drug-coated balloons become mainstream therapies in the coronary field? And this is my answer. The first thing we have to do is stop trying to convince the field that drug-eluting stents are not really needed, because that is not really going to work, and that's not really the intent. Instead, drug-coated balloons need to go head-to-head in clinical situations where drug-eluting stents are actually less desirable. Thrombogenic environments, high bleeding risk, tortuous vessels, lung diffuse disease, and diabetic patients. Also, it is important to know that the clinical conditions actually have changed and technologies have changed. Operators are more comfortable treating more severe forms of coronary disease, including diffuse calcific lesions. Adoption of imaging and physiology guided PCI is progressively increasing, so there is an opportunity to do smarter, angioplasties. And lesion modification technologies have the potential to improve the outcomes of balloon dilatation. And this is important because one of the biggest issues with drug-coated balloons is that at the end, a drug-coated balloon is a balloon. If we can actually, add synergistic tools like lithotripsy, scoring devices, and preparation devices, you could potentially decrease the potential side effects or complications, like, for example, threatening dissections, and actually have better clinical long-term results. I got this actually case from Antonio Colombo because it's really amazing when you actually see cases like this, and I know you have reviewed a lot of these cases during this course. And this is what actually I say, it's time to redefine actually PCI. And I think that actually my three words would be prepare, confirm, and individualize. Lesion preparation in this case is extremely important. We have now a great tool that can actually even fight calcium and prepare the lesions in a way that actually, we don't have before. We have now ways to confirm patency by using actually IDES or physiology to make sure that the vessel is patent after the lesion is being prepared. We have drug-coated balloons with very good efficacy profile on the paclitaxel side of things. And we don't need to get rid of the stents. We just need to individualize the use. When it's needed, and how it's actually used. If you need a stent in certain location, you put it. But the concept of actually, jailing a complete vessel from the tip of the artery to the proximal vessel, at least for me, and as know some of you, actually is not something that is appealing, especially in patients with diffuse and lung disease. And it's really amazing to see how these vessels heal, over time, maintaining, the architecture of the vessel and the functionality, which I really believe is a great attribute of this technology. You were talking about the disparity in use of drug-coated balloons. You see the ratio of DCB TS in the US is still 0, which is actually quite embarrassing. And I really hope as data is generated and clinical trials are generated, we start to move the needle. And I really think that this is actually the best time for drug-coated balloons to really make a difference. But this is the time to invest in randomized studies, in big studies, generate data, and demonstrate the technology really has the value that we all believe they have. So what are the main takeaway points? The clinical benefit of coronary paclitaxel-coated balloons has been already established in specific coronary lesion subsets. I believe paclitaxel-coated balloon offer biological advantages in situations in which drug-eluting stents performance, is suboptimal, such as actually lung disease. However, technique is more demanding and requires the judicious use of ancillary imaging and or physiologic guidance. Drug-eluting stents will always have a role. We just need to redefine when, where, and how to use them. Sirolimus-coated balloons are an attractive therapeutic alternative, and head-to-head comparative trials against paclitaxel-coated balloons are critically needed, and I really want to see the data because actually, biologically speaking, they offer a very interesting alternative. But one big challenge that we have is, yes, data actually is emerging, but drug-eluting stents actually set a very high bar for clinical trials. And if we ever want to actually demonstrate the benefit of these technologies, we're going to have to go and generate a comparable actually level of clinical data. Thank you very much for the invitation. And I really hope you have a great rest of the meeting. And I really hope we continue working together because I really think this field is about to get very, very exciting.
Thank you very much. So I have with me Tuomas Risanen and Teresa Bastante and Felipe Hernandez. You recognize my voice, Juan, it's Patrick Serrois. So we have a few questions, at least I have a few questions, because we don't get very often the chance to discuss with somebody who tested these things in a preclinical state. There is a slide circulating that we see in different meetings of DCB. I saw in Washington, I saw at DCT, and more recently at PCR. This is this logarithmic tabulation where you have solution, everolimus, and then another sirolimus. And it's a logarithmic table because at the top we have something which is 22 nanogram per milligram at 30 days, followed by 60 days 18 nanogram, and I heard that data has been also provided to the FDA for the 90 days. And then at the bottom of the logarithmic 6 we see 0.118 nanograms, so clearly something 100% less at 30 days. What is your interpretation of that? What do you do of these? Are these data coming from your lab or from other lab?
No.
No.
Well, some perhaps was generated in our lab, but no, this is not our own data. But look, you are asking perhaps one of the most fundamental questions in the Limus world right now. Pharmacokinetics in an experimental setting are very challenging to interpret. But there is one thing that we learned with paclitaxel. Despite the variability in the results, despite that actually different technologies showed different pharmacokinetic profiles, the sustainability of the paclitaxel tissue levels were maintained long term and were always above the threshold that was defined to be efficacious for drug-eluting stents. With big standard deviations for many reasons, but that was actually a consistent finding. So that actually gave us a very good sense of security that we could achieve efficacy in humans. With sirolimus or Limus derivates, the story is actually a little bit different because tissue levels actually drop very quickly, and because the variability still exists, but the tissue levels, the total tissue levels as you're describing, in some of these balloons are below the therapeutic level that we used to see with drug-eluting stents. And one concern that actually I have is, you remember the story with the sirolimus drug-eluting stents with the fast release. Limuses are phenomenal drugs as long as you can really maintain consistent tissue levels within the therapeutic effect throughout the entire vessel. Otherwise, actually, you're going to actually not to have a sustained effect in restenosis. So that is one thing that actually concerns me. But again, we have actually seen some interesting clinical data that shows that sirolimus balloons actually does work and does have a biological effect. The question is, compared to paclitaxel products, what's going to be the added benefit?
Okay, so.
This is co-chair Tuomas Rissanen from Finland. Great talk. I would like to ask, how is it in the US discussion between paclitaxel and Limus within cardiologists because of the fears, as you mentioned, on paclitaxel, distal embolization, possible cytotoxicity, even though they are not translating to clinical, endpoints. So how are people feeling about these two drugs?
Look, so you know that because the drug-eluting stent, data, I mean, there is at least a marketing favoring actually the use of Limus, medications. But for balloons, I really think the attitude towards paclitaxel-coated balloons is very positive. But it's sad to say. I mean, we feel like we are in an underdeveloped country because we don't have paclitaxel-coated balloons in the U.S. and no clinicians have access to them. But I really believe they will use them once they become available because it's a real clinical need.
Antonio.
You are Antonio Colombo. Can you hear me?
No, apparently not. Come here.
Yuan, Antonio Colombo. Can you hear me?
I can hear you very well, very well.
Thank you. I have a question. Knowing the attributes of paclitaxel-coated balloons with data, what are the advantages? Why are we pursuing sirolimus? What are we hoping to get from sirolimus? Except for the name.
That's what I said. I mean, I remember when the wave of sirolimus developments started, was at the peak of the controversy and potential toxicity of paclitaxel. So it was created, in my view, mainly because it was actually felt that paclitaxel was a toxic product, based on drug-eluting stent data. But as I put on my slides, I really don't believe that's the case. And the second reason why actually they started, in my opinion, is because the technological assumptions, were comparable to metallic stents. They are not comparable, because in a metallic stent, you encapsulate the drug into polymer, you control the release, the drug is protected and is released over time. With a balloon, you pretty much drop the drug into the tissue, and any level of exposure that the drug has to the environment has a risk to get inactivated by the environment. So technologically speaking, if you ask me, is a more complex delivery system. If you could make it work like a stent, you could potentially actually get a more potent anti-inflammatory effect than paclitaxel. The problem is, in the way they are constructed right now, is a challenging technology. Is actually challenging technologically speaking.
So you mean that if you can make it work, maybe better, but it's only theoretical, maybe better, because why do you need the anti-inflammatory? There is an inflammation when you put paclitaxel?
Yeah, I mean, if you could actually make it work like a stent and maintain therapeutic tissue levels, over time, with that level of certainty, you could potentially achieve the same differences we saw with metallic stents. But remember that actually the concept here is different. And one of the things is the fact that paclitaxel stays deep into the vessel and the vessel is not stented actually produces a biological effect that is very different compared to metallic stents. But my biggest concern, Antonio, is what I really think that people have not really appreciated is that paclitaxel delivery with a balloon is offering a different type of biological profile.
Yeah.
No, at the beginning of this meeting, we heard a presentation from Renu Virmani, and she kind of scared us with crystal embolization and showing the cytotoxicity of paclitaxel. So we are a little bit surprised to see from established people different opinions.
Before going at the response, Juan, we have seen during the meeting here, people are starting to measure IMR after sirolimus, IMR after paclitaxel, and they see some difference, okay? So it fits maybe with the concept of microembolization, but there is so far in larger series no relationship with the classical POCI, nothing. Is there possible that there will be something on diastolic function of the myocardium and discover something later?
That's right, you're wrong. Listen, at the early stages of balloon approval, we did extensive myocardial segmentation and counting the microvessels that were occluded, and Renu reported on it. I mean, there is no doubt that these microcrystalline embolizations actually happen with sirolimus as well. But the thing is, when you actually look at the entire data set in peripheral cases and coronary cases, there are no really signals that essentially show that any differences in major cardiac events. Now, what Patrick is saying is plausible. I mean, is that going to potentially affect diastolic dysfunction and affect, relaxation of the muscle, all these things? I mean, I guess theoretically speaking, yes, but I would actually say with the new generation of paclitaxel-coated balloons, that coating that goes distally mostly actually disappears, mostly gets absorbed, and actually the rest pretty much doesn't have a meaningful effect in myocardial performance.
Very good. We go back to the father of all these things, the person why we are 18 years later still at 5 o'clock in the auditorium. Bruno Scheller has a question for you.
Hi, Juan. Nice to meet you. I have again a question with respect to the United States and in this discussion, sirolimus versus paclitaxel. I fully agree with everything what you're saying, but if you look at it to the US, my impression is that it's much easier to get IDE approval from the FDA if you have a sirolimus program than if you have a paclitaxel program. Is this only an impression, or are there other reasons for this?
No, I think, look, I mean, the requirements will be the same. I don't really think that actually the requirements will be different. I mean, the problem again, as actually I said before, is that the perception of the safety profile of Linus is actually much better appreciated compared to paclitaxel, you know, because the drug-eluting stent data. So I would actually say that actually perhaps it looked with more favorable eyes, but I really think the amount of data that needs to be generated, you know, is pretty much actually the same. But let me tell you one thing, what for me is the bottom line. Sirolimus actually can have a much better safety profile than paclitaxel. Let's assume that actually that is true. I don't believe that, but let's assume that is actually true. But if it doesn't work, and if actually comparative with paclitaxel-coated balloons, if the efficacy profile is actually less, you know, how does that help you? And I really think that's the big question. I mean, Antonio asked the right question. If you actually bring a new technology, in this case, Linus-based technologies, what is the value added? You assume that actually efficacy will be superior, and performance and safety will be much better. Right now, we don't have actually concerns with safety. The question is this going to be able to match the profile of paclitaxel products?
Okay, I think the internet is getting tired because the words were coming slowly. Guan, thank you very much. I mean, we all appreciate for this. You could see that all the questions were very, very important. And I hope nobody will resurrect tacrolimus, because that will be another thing. You remember the tacrolimus, okay, which was much better for the endothelium,
exactly.
It didn't work. Okay, thank you very much, and have a good day, yeah? Bye-bye.
Yeah, you too, thank you.
Okay,
Bye.
so we go back to Adnan Castrati, class effect versus not all DCB are created equal.
Thank you, Patrick. So what is the definition of class in the case of drug-coated balloon technology? Has it to do with the type of drug? Has it to do with the drug dosage? Or is the excipient that defines the class? All of these components might be very important in the definition of the class effect, but it's very difficult to generate specific evidence for each of these components. So that's why I'm taking them all together. Types of available evidence for drug-coated balloon technology. Head-to-head comparisons of paclitaxel-coated balloons. So it has been mentioned here many times, the long list of available paclitaxel-coated balloons. And you see that the differences are in the excipient, much less in the dosage, going from 2 to 3. And there are very little data about direct comparisons between different types of paclitaxel-coated balloons. We have done it in the ESA Desire 3A, which is a prospective study, but it's not a randomized. It's used a historical control. We use the data from the ESA Desire 3 to compare with the data from Agent, which were obtained during the ESA Desire 3A study. You see the numbers here. We got 125 patients enrolled in this prospective study and compared them with 137 patients in the ESA Desire 3. All these patients got the sequent please paclitaxel-coated balloon. You see there is no difference in the diameter stenosis at follow-up angiography between the two types of paclitaxel-coated balloons. And you see some difference in the target lesion revascularization rate in favor of the sequent please balloon, but the difference was not significant. We did the same with another type of paclitaxel-coated balloon, Panthera lux, was used in the ESA Desire 4, which assessed the value of scoring balloon. We took this data in the group of conventional balloon and compared them with, again, with ESA Desire 3 with the paclitaxel-coated balloon from B. Brown. Here also, we couldn't see a difference, a significant difference in diameter stenosis at follow-up angiography, and also in target lesion revascularization. What we have more in comparisons between the different types of particular cell-coated balloons? We have this small study with only 45 patients randomized between Dyer with shellac as excipient and impact falcon with urea as an excipient. This is a study showing that the results with Dyer were worse than with impact falcon. And also in terms of not only lumen loss, but also neo-intima volume. And so this is the only study showing a significant difference. It is a small study, and it is very specific study taking together two particular cell-coated balloons that are not typical for the usual for the use in the cath labs. We have also this study, dissolved versus sequent particular cell-coated balloon, the dissolved IZR randomized controlled trial, which was done in China. I had the chance to review this paper here. You see no difference at all between, and you see also the excipient in both of the coated balloons used in the study. There was no difference in segment late lumen loss, which was the primary endpoint. But if you look at the numbers of target lesion failure and binary restenosis, there is a clear difference which didn't reach statistical significance, but the authors were obliged to add this sentence in the conclusions. Dissolved DCB with its numerical increase in target lesion failure and binary stenosis warrants assessments in larger clinical trials. So if we summarize the the experience with head-to-head comparisons of paclitaxel-coated balloons, we can say that lack of clear differences between different paclitaxel-coated balloons is mostly due to the limited number and size of dedicated randomized trials. And sequent-please paclitaxel-coated balloon is, however, established as the benchmark control drug-coated balloon. Now let's move to the head-to-head comparisons of Limus versus paclitaxel-coated balloons. So I have borrowed this slide from Scheller. He used it in PCR this year. And you see the different types of Limus coated balloons. So what we have only data from the Biolimus coated balloon and also from the sequent Sirolimus coated balloon. We have been eager to know the data from the Magic Touch, Touch. It was expected to be shown at the PCR, but we lost this chance. This is the first randomized trial, Sirolimus versus paclitaxel drug-coated balloon in in-stent restenosis. The trial is also small, 50 patients only, randomized between the two technologies. And what this trial showed, it was published 2019, no difference, no significant difference in diameter stenosis at follow-up between the two technologies. We have heard about the results at PCR from Fernando Alfonso. It is the CVT IZR study, which assessed the everolimus drug-coated balloon in patients with in-stent restenosis. It is a single-arm study. It is prospective, but it's a single-arm study with only 51 patients. You see the results here, no comparison for sure, but it's, they look good with a late lumen loss of 0.40 for in-stent restenosis. It's not bad. And also the clinical results, the clinical outcomes were good. After that, we heard about the REFORM trial, which compared Biolimus with paclitaxel drug-coated balloon in in-stent restenosis. The data were presented by Robert Byrne at PCR. They had randomized about 200 patients between this new balloon and sequent-please balloon, but the results were very clear in favor of the sequent-please balloon. You see that everything quantitatively is in favor of sequent-please balloon. On the other side, there is also another finding which speaks against the Biolimus drug-coated balloon. The positive remodeling, the vessel enlargement, was seen much more often with sequent please than with Biolimus coated balloon. We have also a comparison in de novo lesions. It is a small study done in Malaysia, and Bruno Scheller, who was also co-author of this paper, 70 patients with coronary de novo lesions, not in stent-free stenosis, randomized between the two technologies. Both were balloons from B. Brown, one with sirolimus, one with paclitaxel. There was no difference in late lumen loss between the two technologies, but what was shown there, even here, the lumen enlargement was seen more often with the paclitaxel-coated balloon than with sirolimus eluting balloon. This was the only difference. So back to balloon, I don't want to lose much time with this. We have seen a lot of cases here, but this publication is interesting to show the dissection in OCT and the healing at follow-up, which is beautiful. Also, I don't need to tell much why paclitaxel might be superior, because it was dealt with from Granada also now, and before from Virmani. And I want to conclude with very provocative conclusions. Please keep the calm. And I'm not trying to discredit a technology, but just I want to encourage more intensive work in that. So I'm saying here it took a relatively short time to understand that paclitaxel was inferior to Limus drugs for drug-eluting stents. It is taking a much longer time to understand that paclitaxel is superior to Limus drugs for drug-coated balloons. Thank you very much.
Okay, thanks. Do we have questions, or do we continue at this point?
Yeah, just a short remark. I mean, the people were very optimistic about the Biolimus A9 because it was said classically that it is 9 times less hydrophilic than the sirolimus. So they thought, okay, that's something lipophilic, and they will stay long in the vessel, and they have to recognize that it was not the case. I think we can.
Yeah, I can. Let's continue because of time restrictions. So the next talk is given by Raban Jäger, and the title is Different Available Devices.
So I think you will see me for the last time today. I have a little device parade. Some slides are similar as the slides Professor Castrati showed. These are, this is the zoo of interventional devices, drug-coated balloons we have at the moment. There have been some more added because this is the coming out of the publication of the consensus paper. And I just picked some of those with some data. We have seen data partly already. The Elutax SV balloon is a balloon that is covered with paclitaxel. You see here in the middle the balloon. So this is the balloon, and outside there's a layer of ice and then snow. So this would be a good balloon for Finland, I think. And outside there's a seal layer of dextran, and the company says this protective hydrophilic sealing of dextran is giving much better mechanical stability of the drug coating, seems to be anti-thrombogenic, and is really putting the drug where it has to be during inflation. This is what the company tells us. This balloon has been tested in the Picoletto trial, Picoletto 2 trial from Bernardo Cortese. We have seen this slide before for a short moment. This Picoletto 2 trial was a randomized non-inferiority trial that tested this Elutax SV balloon against science and in small coronary vessels. And The primary endpoint has been angiographic, but here you see the 3-year clinical outcome, which was really favorable for the DCB. And so I think this concept of this balloon might be working. When we continue with other paclitaxel balloons, you see here the Sequin Please, the comparator of the DCBs nowadays, as we heard. This is the classical iopramid matrix coating with the paclitaxel drug, and this has been tested in various trials. Of course, I show my basket small 2 trial, but many other trials has tested this device, and this device is the best documented device on the market at the moment. Another interesting device is the Restore balloon from Cardiología. This is again a paclitaxel balloon with the same amount of paclitaxel added. The excipient is shellac ammonium salt, and this is something the company is calling SAFE PACS, or the SAFE FE PACS technology. I don't know how they pronounce it. And this device has been tested as well in a large clinical trial, not that large, but several hundred patients. The Restore SVD trial, I already showed you this slide, and the 5-year clinical data is good here. So this is, again, a balloon that seems to work. Then we switch to the Limus. The Limus, as we heard, is a very appealing field. Lots of trials going on. Everybody jumped on these balloons. The Magic Touch balloon from Concept Medical is working with a technology they call nanolute, and behind this nanolute are these submicron particles of sirolimus that are encapsulated by these phospholipids, and these little balls, they are hanging around on this balloon, and by inflating the balloon, these little balls go to the wall of the vessel and are getting a part of the vessel wall, because the phospholipids are the same in the cellular wall. And so this is the concept of this. Looks quite interesting. Transform 1 was the trial that tested these Magic Touch balloons against the Sequin Please in smaller vessels for the 6-month net gain. And as we heard, this trial was scheduled for the late breaker at EuroPCR, but has not been shown. And so we're still waiting for the data, but we are really interested, because this, if this works, this could be a good balloon, and if it's not working, it's not a good balloon. But we're still waiting for the data. Another sirolimus balloon is the Solution balloon. They work with this so-called micro-reservoir technology and the cell-adherent technology. I've been diving a little bit into it, but I'm still not understanding it. But no matter, apparently this sirolimus is encapsulated in this biodegradable polymers and is somehow protected by this cell-adherent technology, in order that when you insert the balloon, this material doesn't get transported anywhere. So it stays on the balloon, and by inflation it gets to the vessel wall, and there it has a sustained release of the drug, in order that several weeks the drug is working there. So as far as I understand. Well, we don't have any direct comparison of this Solution balloon against paclitaxel balloons, but we have a large clinical trial. Simon Egglesall is the PI of this study, the Solution de Novo trial, that is enrolling throughout Europe. Interestingly, this is trial that is randomized before vessel preparation, so this is not what we heard about the whole day, but it's testing a strategy of DCB against DS, allowing a hybrid DCB approach, and with this trial we for sure will have some interesting data. And then the sequent SCB, we have seen this slide before, the same late lumen loss in direct comparison to the sequent please, a little bit less. The rate of positive remodeling is a bit less in the SCB group than in the paclitaxel group, but there is some positive remodeling. 1/3 of the patients they have it. It's not 2/3. It's not 2/3 of the patients. It's just 1/3. But at least there is some positive remodeling here. And in this direct comparison, this angiographic comparison, the outcome was quite good. And the last device I want to show you, we have seen that bioluminescence-coated balloon, the Reform balloon. This talk has been presented at EuroPSA by Robert Byrne, and here you see this is another representation of the results. You see that the biosensus balloon has been a little bit worse than the sequent please in this direct comparison in stent restenosis, and so we wait for the next generation of this balloon. So I will end with the PCR statement. There has been a PCR statement issued on May 17th, and they say that 13 paclitaxel-coated balloons are around with various formulations, drug formulation, excipient formulations. There are 4 sirolimus balloons and 1 bioluminescence-coated balloon. And at the end, there is no, as we heard it from Juan Granada, there is no balloon approved in the US. However, there are 3 balloons they applied for an IDE study. This is the agent, this is the solution, and this is the magic touch, and all these applications, as far as I know, are still running, but the trials have not been started yet, but I'm not sure about this. And we heard about the class effect that is not there, and every device actually has to be tested against the comparator, and we still wait for various data, and I think the future is very interesting, because more possibilities we have, the better we can treat our patients. Thank you very much.
So we will have the question later. The only thing I would like to say is that the solution, you have this a little bit mysterious cat, yeah?
Yeah.
Because I have heard of, from John Schulz, which is the brain behind, that there is also something with electrostatic charge.
Yeah.
I didn't know that the 70 mi is more positive or negative. I forgot the detail. Do you know something about that?
No, but I think it's, I don't know anything about it, but I think it's really essential that these little elements, they stay on the balloon during transfer into the vessel wall. I think this is essential. When you lose all this material during passage, nothing gets to the vessel wall, and I think it's apparently it works, but Simon can tell us more about it.
So what has been... oh, sorry.
But
the only point before you respond is the other point. I was talking to Jeff Jump. Apparently, it's a strategy trial. I mean, you are not penalized if you put a stent in the patient. And so far, the rate of balloon of stent is about 20%. Okay, and it seems to be stable now at 20%. So that's quite interesting, because it's a kind of provisional
totally agree, yes.
Yeah, provisional balloon, and then I asked them if he had an idea which patient go to the stent. They say that they have a session every week of 2 hours to review this case, And I said, yeah, but what kind of dissection? He said, oh, we don't have 5 classification. We have the classification B and S, balloon or stent dissection. So we still don't know at what moment they go to the stenting.
Well, I think it's the decision of the investigator or the interventional cardiologist, and so it's really based on the angio.
Thank you, Raban. And, the next talk is by Bruno Scheller, and the title is Limus DCB Ready for Prime Time.
Okay, I will have to shorten my presentation a little bit because there's a lot of overlap with the prior presentations. Before I give my statement about Limus, I want to again say that the reason why we are talking about Limus for DCB is not due to the fact that paclitaxel is not safe. That's very important. In my opinion, there is a big need for Limus-coated balloons because the perception in the community for Limus is much better than for paclitaxel. And in my opinion, part of this strategy to come out of the niche for drug-coated balloons is to get a better perception in the whole community, and maybe Limus may be a tool to get a better product acceptance for this. So this has also been discussed. We have different mode of actions between sirolimus and paclitaxel, and what has been said before, the big advantage of paclitaxel is that it has an irreversible binding to the cell. This means it goes into the cell, and this cell is blocked for the rest of cell life. And that's the big advantage, because for Limus you have reversible binding to this mTOR receptor complex, and this is exactly what has been discussed. For Limus we have to guarantee sufficient drug persistence in the vessel wall over a long period of time. Last word to cytotoxicity. Much of this discussion is based on this paper from your group, Adnan, the Wesley paper from, 2007, where you looked at cytotoxicity and apoptosis with sirolimus and paclitaxel on smooth muscle cells and endothelial cells. And what you can see is that cytotoxicity is almost always the same for sirolimus, independent of the local concentration. And this is the reason why we say there's a much broader therapeutic window. For paclitaxel it's different. There's a dose-dependent cytotoxicity, and beginning from tissue concentrations of 100 nanogram per milligram, paclitaxel gets more cytotoxic than sirolimus. Below this threshold, it has a very similar cytotoxic behavior as sirolimus. So, and the important thing is that if you have paclitaxel from the stent struts, then you have a very high concentration nearby the stent struts, and then it's very possible that you exceed this threshold for cytotoxicity. Balloon-based, we have a very homogeneous drug delivery, and here, if you look at this paper from Gongora for the FDA-approved paclitaxel peripheral DCB, you see that after 4 hours, you do not reach this threshold of 100 nanogram per milligram. And then it goes down, and we are far away from cytotoxicity. Therefore, the claim that paclitaxel is cytotoxic in combination with DCB is, in my opinion, somewhat misleading. Okay, I will skip this. This has also been shown before, the different distribution in the vessel wall. Paclitaxel has a higher concentration in the adventitia, and that may be one of the reasons why we see this effect of lumen enlargement more frequently with paclitaxel than with sirolimus. There were many projects in the past. For example, we had also a project more than 10 years ago together with Abbott on sotarolimumab. However, the required concentrations on the balloon were too high, and therefore they stopped this program at this time. This is the slide from PCR on the current landscape, at least the programs we have some information about. There are many more programs to my best knowledge at the moment, but for these programs we have somewhat reliable information. This is a slide we created several years ago about the preclinical data of the different programs. Solution is not here, but the other programs are included. And you see different acute transfer of the sirolimus in the tissue. However, we have to be aware that these values depend more on the preparation and analytic technique than on the real drug transfer. So it's very hard to compare these data from different experiments. But if you say, okay, they have all a very similar acute transfer, and we look at different time points, then we see that, for example, for the Magic Touch, there's almost no drug anymore in tissue at 4 weeks, whereas other concepts, such as the highly crystalline coating, show respective drug concentration after 4 weeks. In my opinion, the most potent tool for limus drug delivery is the virtual balloon. This is not a drug-coated balloon, this is a drug delivery balloon, because it allows to transfer much higher drug concentration than it is possible with DCB. So I think with this tool, at least theoretically, it is possible to reach high enough concentration in the tissue and maybe persistence to have a biological effect. The disadvantage is, however, that this is a delivery tool, and for example, if you have side branches in this area, it's hard to control where this drug goes. There is a published registry years ago, 50 patients in ISR with respective values, but no randomized data. This is Magic Touch. We already talked about the rapid decline in tissue concentration, which may be a severe limitation of this device. And we have talked about Transform 1. It is not presented, unfortunately. However, Bernardo gave some information on interim analysis, and it will be interesting to see the final data of this. There's a larger trial ongoing, the Transform 2 trial, with more than 1,000 patients against drug-eluting stents, and I think this is a very important trial for this device. Solution has also been shown before. We have here the tissue concentrations published by Thomas Zeller with not a logarithmic scale, with a normal scale. And there you can see that it starts with a high concentration and has some persistence over time, up to 60 days is presented here. Simon will report about this largest ongoing trial, so I will skip this. For the bioluminescence balloon, I was honestly disappointed to see this data. I expected a non-inferiority because the first data published on this balloon in small vessel disease, the BioRise China study, were not that bad. They included more than 200 patients with small vessel disease, randomized 1-to-1 to the bioluminescence balloon or POBA. And as you can see, there was significant benefit by the bioluminescence coating. Late lumen loss was 0.16 versus 0.3 for POBA. Nevertheless, this is a very good value for POBA, I think. But it was clearly significant and showed the efficacy in de novo small vessel disease of this type of balloon. If you look at lumen enlargement or positive remodeling, it was the case in 10% of the cases for POBA and 30% for the bioluminescence balloon, which is in line, which has been reported for other devices. Reform has been shown already, was a negative trial with also the bioluminescence balloon in instant tree stenosis. However, this was, to my understanding, a different coating that has been used the BioRise China study. What we did years ago is we looked systematically at different formulations for sirolimus on balloon coating, depending on crystal modifications. So we tested completely amorphous coatings, mixed coatings, and highly crystalline coatings. And interestingly, a crystalline coating with a sirolimus concentration of 4 micrograms per square millimeter balloon surface had very good drug transfer, and what was very interesting, that we saw about 50% of the drug after 4 weeks still in the vessel wall. So this was, from this perspective, a promising coating, which has then been tested in first mechanistic clinical trials. The first ISR trial conducted in Malaysia, 50 patients, 1-to-1, randomized to the conventional paclitaxel-coated balloon, the sequent please, versus the highly crystalline sirolimus-coated balloon. Typically for these studies in Asia, patients are much younger than we see it, about 60 years on average, but a very high percentage of diabetes in these populations. And this was the QCA, primary endpoint, lane lumen loss in segment, was not different between the two groups, 0.21 for paclitaxel and 0.17 for the sirolimus-coated balloon. Clinical events not different, but not enough patients to draw any conclusions. What we also typically do, the same as with Pacocat in the beginning, we conducted in parallel a second trial with the same design, but this trial was conducted in Germany and Switzerland at Ravans Center. Same design, same number of patients, so at the end we had, in this pooled analysis, we had 101 patients, 1-to-1 randomized, and what the European patients were 10 years older but had less diabetic, so this is the difference between the populations, the main population, and what was the outcome was interesting. In the Malaysian population, there was numerically an advantage for the sirolimus balloon, whereas in the European population we saw quite the opposite. And at the end of the day, the matched population was more or less equal with respect to the primary endpoint, late lumen loss in segment, 0.26 for sirolimus and 0.25 for paclitaxel. And this is the late loss distribution, here the MLD distribution curves. Clinical follow-up was also not different. And the third published trial from this series is the de novo trial conducted in Malaysia, 70 patients, randomized 1-to-1, again sequent please versus the highly crystalline sirolimus coated balloon in de novo disease. And this is the main finding what has been presented before. Late lumen loss in segment as primary endpoint was 0.00 for the paclitaxel coated balloon and 0.10 for the sirolimus coated balloon. The non-inferiority criterion was fulfilled, so this means the sirolimus coated balloon is non-inferior, but there was this difference in late lumen enlargement, 60% with paclitaxel, 32% with sirolimus, and this compares very well to the BioRise China study, which also saw an increase for the limus drug for 30% of the lesions. POBA 10%, paclitaxel 60%. This is the ongoing study program. We are in the final phase of analyzing the European de novo trial, which will be presented hopefully later this year. There's a large registry ongoing, and we are planning, under the lead of Raban, a large de novo trial, which is called Basketball at the moment, as working title. So hopefully we can start with this trial later on. So as said before, paclitaxel-coated balloons are still the standard because we have much more data than for sirolimus. And all the technical issues for sirolimus-coated balloons have been discussed. And the main limitation at the moment is that there's very limited published clinical data. We have mostly registry data and only a few small randomized trials. And therefore, it is important that we do serious clinical science in this field and do not repeat the same mistakes we did with the paclitaxel-coated balloons in the beginning. Thank you.
That was really a tour de force. I mean, could have summarized the whole day in one shot. So many information. I hope that we will have a video to digest that, because there are, of course, many questions, but we are late. We go for the last one.
Yeah, Simon has the privilege to close this session with ongoing studies with Limus DCB.
Great, thank you. Not very often you get the chance to follow Bruno talking about DCBs, is it? And it's a dubious honour, I think, is probably what I would say at best. So I wasn't really sure quite what I was going to be talking about with the ongoing studies. So I've got talks that sort of are pretty much the same as Bruno's and Raban's in some ways, but maybe we could talk about them in a slightly different way. So I have to say thank you to a lot of people that have given me some or all of these slides. There's one of Bruno's for a start, making the point that all the balloons are different, essentially. So I'm very briefly going to talk through these. I don't know whether, bearing in mind that we're overrunning, we could have questions during rather than after, I don't know. So this again is from Bernardo's work with Magic Touch. That's the study that we were all waiting for, which currently is under embargo, I think, while the data is being reviewed, which does sort of question what they're going to do with the next study, but I gather that's ongoing. So this one is a important big vessel, large number of patients. Randomization is after lesion preparation, like quite a lot of the studies are. So the solution study is, well, the solution balloon has been shown already by Bruno. This is the trial that I'm involved with, which is quite ambitious, I think it'd be fair to say. 3,300 patients, randomized 1-to-1 against whichever current generation drug-eluting stent you happen to use in your department. It's all comers with the rather obvious exceptions of left main stem, STEMIs, vein graft interventions and CTOs. So we hope that it will be as big an all-comer study as that you could have in one go. I quite like the fact that it's randomization before you start doing anything. So the idea is you take your angiogram, as long as the patient fulfill all the appropriate inclusion-exclusion criteria, you then have to decide in your own mind, is this a patient I can fully revascularize with all stents or with all DCB? And if the answer is yes, then they go in the study and then you're stuck in that arm. So if you're doing multivessel angioplasty, for example, and you're in the solution arm, if you bail out on the first lesion that you treat, they remain in the solution arm and the rest still remain solution rather than stent. I'm sure there'll be lots of discussion about that afterwards. This is enrollment, currently something over 1,200, about 1,100 of those in the last year. I'm pleased to say that anywhere in the coronary tree is being treated. The balloon size and length, it looks like an all-comers study. We've got over 60% or over 3 millimeters in diameter. Bailout rate, as you said, is around 20%. We're trying to have to do things like work out as we go along that actually it's not just bailing out. So you have to classify it as it's a failure of lesion preparation. So you've crossed over to a stent, but we don't call it crossover, we call it bailout. Or you bail out after you've used your solution balloon for those rare instances where you've made things worse rather than hopefully left them the same. So the vast majority of this will be a failure of lesion preparation. So this has already been mentioned, the Biolamus study. These are the slides from Keith Oldroyd, but from Robert Byrne. So I had a chat with Keith after PCR because I knew we were going to be talking about this. So I thought, he's the CMO of biosensors. I thought he might have some insight as to what he thought was going on here. So as you know, this is the first generation Biolamus balloon that they've used. And the bit that's highlighted in red is the bit that probably is what they think is causing the problem. It's a crystal size, which is quite a spread, but the average seems to be coming out about 10 microns. And they started their clinical trial in 2019, and I think some of the confusion over crystal size and the recruitment for the study was hit by COVID, like so many other things were. So this is what they're doing. They're enrolling 2 to 1, as we've discussed earlier. They missed their non-inferiority, we know that already. One of the interesting things is how much positive remodeling you seem to get with the B. Braun sequent please balloon in an ISR study, and I'm not sure that's been seen before, is it? And I can't explain it. I'd actually asked Keith quite how they did this, what their QCA was, to see whether it's actually something to do with stent expansion or plaque regression within the stent. I'm not sure they know the answer to that yet. But one of the things that does come out of it, I presume because of the amount of gain that you're getting inside the stent with the sequent please, that the overall late lumen loss is really quite low compared to any other studies at 0.2. There's a number of other issues around that Keith was highlighting to me, which one is the difference between looking at 6 months and looking at 9 months. And I'll be interested to know from people here whether they think that makes a significant difference. And also there's the ongoing discussion about, do you use late lumen loss or do you use diameter stenosis? So in China, they're using what they've now decided is their second generation Biolamus balloon, where the crystal size is more like 1 to 3 microns rather than 10. And they're saying that that gives a double the tissue penetration of the drug. So that may well explain the problem. So their in-stent restenosis study over in China with 280 in each arm is using that. It's completed and they're awaiting the outcome of that. So this is all Bruno's work. I've taken out most of your slides, Bruno, because I thought you might be using them. Just to say that I thought this was really impressive. The fact that your current sirolimus balloon is performing as well as probably the best paclitaxel balloon on the market currently. You've seen that again as well already. So the very last one, which Fernando very kindly sent me the slides for, is the everolimus first-in-man study, which you have already seen as well today, which showed good primary effectiveness outcome. So where are we now and where do we think we're going? So I guess one of the most important things is we're waiting for the Transform study to actually report so that we know what's going on there. I'm pleased to say with Solution it's up and running and it's recruiting fast, which is good. The Biolamus balloon, I guess they need to work around their second generation concept and with their in-stent restenosis study. I don't know what's happening with the sequent balloon. I'd love to know. And everolimus, Fernando informs me that there is a much larger study being planned. So I think one of the things with all these limus studies is what should they go against? Should they be going against paclitaxel balloons? Should they be going against stents? My view would be, I think the argument really is about the fact that there are too many stents being put in and not enough DCBs. So I would go for stents rather than balloons personally. What endpoints you're looking at? Is it the early angiographic? Is it the late clinical? If it is angiographic, is it 6 months? Is it 9 months? Is it late lumen gain? Is it late lumen loss? Is it percent diameter stenosis? And I think one of the really important things for all the limus balloons is how they think they're going to get around the late lumen gain that paclitaxel keeps on delivering. Thank you.
So I think that what was remarkable during the whole day is a few slides were presented multiple times. But that's a good thing. That's a good thing because I used to say repetition is the mother of education. Repetition is the mother of education. And by now you should know all the numbers by memory, you know. How many make me program on this one, on that one, etc., etc. I think if there is no burning question, you have a burning question.
I have one, maybe for Bruno. Do you see any use of combining limus and paclitaxel on the same balloon, or does it just increase the likelihood of something bad happening? I'm referring to my couple of cases where there was restenosis and multiple stent layers, and I combined two drug-coated balloons there just to hope that the patient is not getting back.
Combined coatings is, in my opinion, a very interesting topic. I personally do not think necessarily it should be paclitaxel and sirolimus. It could be maybe a combination of something anti-inflammatory, which together with something which promotes plaque degression, for example, or regression. So the interesting topic, but I think from a regulatory perspective, it's a mess because you have two drugs with their interaction and all these things. I think that's not that easy.
I have a question for Bruno. Among the paclitaxel coated balloons, anyone has an amorphous technology or all crystals? And if they are all crystals, there is any specific crystal formulation which is less likely to embolize or more benign?
Good question. In the beginning we didn't talk about crystalline or amorph because we had no idea what this is and what it means for the coating. So later it turned out that a mix of amorphous and crystalline or more crystalline has a better PK, better persistence of the drug in the vessel wall. That's what we have seen, and the amorphous coating were inferior with this regard, and therefore, to my best knowledge, all the coatings on the market that work are more crystalline than amorphous. So that's what I can say. But it's not only this question. There are other parameters influencing the quality of the coating.
Very good. I think the session is closed, but there's still some exciting case, and at the end of the day, that's the most digestible type of information that you can still receive, because the heroes are tired. They said they have no question. My co-chairman has no question. I have no question, so we leave the scene. Yep.